Can you hear me? Good morning, everybody. For the sake of time, we're going to start. It's 9 sharp. So welcome to the first part of the session on climate change and cumulative effects on ecosystems. So this first part is on the ecosystem reorganizations and marine dynamics. There will be further talks in the afternoon on cumulative impacts. And there's also a session, there's a session this afternoon on Central Arctic Ocean ecosystems, and tomorrow morning on cumulative impacts. As you know, climate change is exacerbated in the Arctic. It's 3 to 4 times faster there than the rest of the planet. Some of the main consequences are loss of sea ice, warmer temperatures in the air, but also in the oceans. And of course, these changes reorganize the ecosystem. The ecosystem have to adapt. It changes. We have polarization. We have new species. We have retraction of some of the habitats. These are further exacerbated by the cumulative impacts from anthropogenic activities. As you know, there's more human activity also cumulative to these natural or climate changes. And there's also, even though there's a trend towards a warmer Arctic, less sea ice, there's also the climate and natural variability. And disentangling these effects is not easy, especially that these changes are not the same in the Canadian Arctic, in the European Arctic, around Svalbard, in the Central Arctic Oceans. So we've selected talks that try to address these different questions and also from different areas, and we hope that you'll have a good session. This morning, we will have talk that cover circumpolar, a good session. This morning, we will have talk that cover circumpolar changes and borealization, ecosystem changes in the last ice area, identification and fisheries and acidification, and harmful algal blooms. Without further ado, I'm going to pass the mic to my co-chair, Edna Erickson. But just before doing so, I would like to remind the speakers that we have 10 minutes per talk. So we know it's very short, and we apologize for that. But that was the way of having that many speakers. And we're going to show 3 minutes, what 7 minutes in your talk, at 2 minutes, so 8 minutes in your talk, and then at 0 minutes. So I'll have the little sign, and I'll be sitting there. Oh, and there's time right there, too. Thank you. Anna?
Thank you, Max. We start our section with Bérenger Husson. Can you put on the screen? Bérenger Husson, can you video? The key speakers. Yeah. Hey, Bérenger. Bérenger is a researcher at the National Center of Scientific Research in France. She studied how climate change is reshaping Arctic marine ecosystem. And in her talks, the spillover borealization of Arctic shelf ecosystem, she explored how warming oceans are allowing boreal species to move north, changes Arctic food webs and ecosystem dynamics along the way. Please, Bérenger, nice to meet you.
Nice. Thanks, Elena. I'm going to try to share my screen, and I'm going to also, please tell me if you don't see anything. No, that's not that. Near this one. And please tell me if you don't see the screen in full size. It's a bit slow, sorry, it's going to take a bit of time. Yes, here it is. Can you see it? Fantastic. I see the backup, which has never happened before. First, thank you very much, Elena and Maxime, for selecting my talk for this opening. I'm very pleased to be able to share the research that we published a year and a half ago or so, something like that, about one of the well-known impacts of climate change on the Arctic ecosystem, and Maxime talked about it, the borealization. I'm very sorry I couldn't be here. I'm trying to reduce my travels, and also I'm a bit sick, so it's better for all of you maybe that I don't spread my germs everywhere. So I'm just going to start right away with what is the borealization exactly. So borealization is a process when ecosystems that were historically characterized as Arctic are progressively or transitively acquiring features typical to more southern boreal ecosystems. So there was a lot of literature on that already, but what we were exploring with this review paper was what was the geographical extent, the biological extent. We wanted to know which functional groups were concerned by borealization, and the functional reach in terms of functioning of the ecosystem. So just to have the same basics on what we call Arctic and boreal, let's say that temperature in the Arctic is year-round in the Central Arctic Ocean or seasonally in the shelves at or near freezing point. The stratification in the Central Arctic Ocean is very strong, with a fresh, cold Arctic layer that is on top of a warmer, saltier one. That is from the Atlantic, and also this fresh Arctic layer is what allows the production of sea ice. I'm just going to add a pointer here. On the boreal side and offshore ecosystems, you have, on the contrary, higher temperature, saltier, and less stratified waters. In terms of primary production, so you have the latitudes here in the y-axis and the months of the year in the x-axis. And you can see that in the northernmost ecosystem, you have first a bloom of ice algae under the first year ice, and then a bloom of phytoplankton, mainly diatoms, that appears after the melting of the first year ice. And then in the boreal ecosystem, you have a longer production period and also a higher primary production. In terms of biotopes, they are quite different too. On the pelagic Arctic, you have a low diversity with big zooplankton, lipid-rich, large zooplankton, and some large fish species, mainly polar cod most of the time, or in some places at least. In the demersal compartment, you have small fish like eel blennies, eel pouts, scotids, sculpins, things like that. And the benthos are like mainly worms and mollusks and arthropods and echinoderms. On the boreal side, you have a much more diverse pelagic compartment with smaller zooplankton, more diverse fish species, and a zooplankton area, compartment that is a bit more diverse, although it has been now recorded also in the Arctic. The demersal fish are mainly large predatory or generalist fish that are very mobile, like Atlantic or Pacific cod or walleye pollock. And the benthos is larger individuals and more arthropod. So this borealization, it's driven by local change in the habitats, in the conditions of the Arctic and around the Arctic, but also by the advection through those red arrows here of warm, salty water surface that are surface water that are carrying a lot of species, but also changing the habitat locally for other species. And that has been shown that these borealization, they are stronger, or you see more signs of boreal species during the years where you have anomalous advection events. So it's from year to year, a cold year versus a warm year. It has been shown for demersal fish, first and foremost in the community, but also a change in the zooplankton community, notably towards smaller, less lipid-rich animals. And also in the benthos compartment, although with a 7-year lag. And this has, of course, impacted the upper trophic levels with a shift among seabirds communities that are shifting also in diets. So that's the borealization that we have mostly heard of. But what we have noticed in this review paper is that there's also a number, quite important, of literature that shows that there are other impacts on other shelves. So I'm not going to show all of them, but I invite you to read about that. There's a lot of elements in there. So the Barents Sea and the Bering Sea, which are these inflow shelves, have the highest warming rate since the 1980s in winter and autumn for the Bering Sea. But when you look at those summer SST temperatures since 1950s, you can see that there's also a consequent or a substantial warming both in the Canadian side and the Russian side. And this is very correlated with the trends in sea ice concentration on the last 50 years. So there's also a strong change in the environment for those other shelves in the Arctic. So what are the potentially changing borealizing communities? Well, when you look at microorganisms, including phytoplankton, there are signs of small algae being more numerous in Canadian Arctic archipelago when the temperature are warmer. Those and the Arctic fresher, those are more efficient algae that are more efficient in oligotrophic environments. And there's also signs of coccolithophores and other temperate phytoplankton species expanding through the Fram Strait into the Arctic Ocean when the years are warmer. In terms of zooplankton, you have smaller and less fast zooplankton in the Disco Bay, Western Greenland, and also in the Labrador, Eastern Greenland, and Fram Strait, you have signs of more Calanus finmarchicus and smaller zooplankton from the boreal environments. In terms of fish species, you have more and more southern fish species like the Pacific sand lance or the capelin, which are growing in numbers in the Canadian Arctic archipelago or in the Hudson Bay and in the northern Baffin Bay. For the musselfish, there are a lot of reports of changes in the community of the musselfish across depth from 300 to 1,000 meters depth. And it has been linked apparently to from genetic study to populations from the Barents Sea that expand towards the west. And on the Russian side, there are also changes with species from the Atlantic that are expanding eastward and species from the Pacific that are expanding westward. So it's really reaching a lot of groups and a bit everywhere in the shelves. So that's one point that we notice from the rich literature that there is about that. And then to go further, please tell me if I'm running out of time. I had scheduled for a bit more than 10 minutes, sorry. For the ecosystem impacts on the functioning of the ecosystem impact, of course, you have the change in habitats, which is most obvious for the marine mammals, including killer whales that are more and more seen in the Chukchi Sea. In eastern Greenland, there is an increase in the abundance of several boreal species like humpback whale, fin whale, killer whale, and pilot whales, but also dolphins. And the sea surface temperature are increasing and also linked to seasonal displacement of bowhead whales in Baffin Bay. Another process that is directly linked to the change in environmental condition is the primary production with the Barents Sea that has been increasing most than anywhere else. The production there has been booming. That has been shown a lot of time. The primary production has also expanded in special extent and production in the Bering Sea. But we also see signs of spatial temporal change in primary production in eastern Greenland and Canadian Arctic in Baffin Bay. It's also impacting fish condition and feeding, including capelin for the Barents Sea, for example, that has a poor feeding and condition and growth. It's also affecting survival, the change of zooplankton, because those zooplankton-rich in lipid that are occurring usually in autumn, before the winter, are a very important source of reserves for all the species that have to survive through the winter. So it is mainly impacting capelin and walleye pollock. So their survival is a bit hampered by this change in communities. On the other hand, as I said, the advection of warm water is also bringing a lot of species a bit more often, including krill. And this krill is kind of stabilizing some of the predator populations and changing a bit the trophic controls there. And with these upcoming species, the boreal species, you have also an increase in competition among the fish species. All of this is changing the trophic food web, especially in the pelagic compartment, leading to an increased food web connectance and decreased modularity, which is associated with a low resilience of the ecosystems. Last few functions, the apex predator conditions and all the red listed species are having poorer conditions or are failing recruitment events. In terms of biodiversity, there was a Pan-Arctic study that showed that the biodiversity and especially the species richness has been increasing all over the Arctic due to these incoming mesopredators, so mainly the mussel fish species. And this is consistent with more local studies that have been showing increase in biodiversity during warmer years. Finally, to the final functional aspects, the functional diversity is also changing. In the Greenland situation, the functional diversity is decreasing because the incoming boreal species do not compensate for the loss of Arctic traits. Whereas in the Barents Sea, the species richness, or the functional diversity, sorry, is increasing, and that's changing the functioning of the ecosystem. So to wrap it up quickly, I showed you here some of the papers that were reporting change in the borealization of communities, or the changes in processes, ecosystem processes, and we can see that it's happening all across the shelves, and this is due mainly through transport of new species, through currents, or through the movement of mobile species which have newly suitable habitats in other areas. But we do also highlight that there's a lack of data for some areas, including the North Russian areas, lack of data for the winter also, and for some groups. And this I would only cite the microorganism, but there's also a lot of other groups for which we haven't enough data to say anything. This affects all functional groups and many ecosystem processes, as we've seen. And there were also other points that we discussed in this review paper. We discussed with the experts that were co-authors what could be the future of the Arctic. So it might continue pushing the border between boreal and Arctic species and Arctic ecosystems. But it's going to stabilize at some point because there are some elements in the Arctic, like the photoperiod or the light and the depth, that are going to be unmovable frontiers to the displacement of species. So this is not going to be lasting forever, and there might be transitory, more borealizing years where you have more water or seasonal changes that bring more warm water, and that will lead to temporary borealization. We also tried to discuss the risk, but of course it's kind of difficult when you don't have any data on species adaptive capacity. This is a big point that is starting to emerge. And of course, there is a consensus that ice-associated species are most at risk. And the changes for this species and the rest of the ecosystem that we've seen has implication for the local communities and the potential for brook growth in the Arctic. So with this, I thank you for your attention. I hope I hadn't taken too much time. And if you need to contact me, don't hesitate to reach me on those links. Thank you very much.
Thank you very much, Berencher. We will see one question. Sorry, technical problems.
Yes. Yep. Questions?
Yeah, Bérangère, we don't see you, but I hope you're online. This is Benjamin Planck from IMR in Tromsø. I was wondering how you dealt with the proper attribution of the changes that you described in this review to climate change, the physical borealization of the ocean. And I was particularly interested or concerned with the publication bias, that many papers may actually report changes but not report lack of changes, and also to what degree there is a consensus in the community on the changes you report. For example, in primary production in the Barents Sea, which is notoriously difficult to summarize at the scale of the whole shelf. So yeah, a little bit on uncertainties, bias, and attribution.
Yes, thanks, Benjamin. Ooh, there's a bit of a echo. Yes, very important question. I have been actually wondering on how to really display that. What I haven't maybe stressed enough in the presentation is that the thing that I highlight in that is happening on the shelf is usually some things that are linked with studies that look at warm year versus cold years. So it's oh, you see me. I have to be careful with what I do now. Yeah, so it's often it's not like long studies that are really treating borealization or per se, or don't say the word really clearly, but they show that when you have warm year, you have advection or development of such and such species, and often they link it to borealization or think that it's borealization, but of course there might be a bias here of whether they want to link it to the word borealization because it's linked to something that people will recognize, or if it's something that, or maybe I can stop sharing my screen. Or maybe it's something that, yeah, that links, that explains a bit what is happening. Also, yes, I have been using the literature that was published for the Barents Sea and the primary production. There was actually a number of papers also by Arrigo and Ardina, etc., for those areas that were not based on the same data. So and it's one of the only areas for which we have a lot of publications. And of course we know that there are uncertainties in those data. That's something that we might have stressed a bit more in the discussions of the paper. What was the other question? I lost track.
We have no time.
I think we're good, Bérangère, but thanks a lot for your presentation and your answer.
Yes. Thanks a lot.
Thank you, Berencer. Next, She will present first insight into the marine ecosystem of Tuvayutuk's fjord in the Canadian High Arctic. Please, Lisa.
Everyone. Yes, my name is Lisa Mattis. I'm from Fisheries and Oceans Canada, short DFO. And I would like to thank the session chairs for giving me the opportunity to take you on a journey to the majestic fjords of the Tuvayutuk Marine Protected Area in the Canadian High Arctic. And the work I'm presenting today is part of the DFO-led Arctic Core program, but it's also a product of many collaborations. So Tuvayutuk means the place where the ice never melts in Inuktitut, and it's located off the northwest coast of Ellesmere Island in Nunavut. The MPA was established in 2019 to protect the multi-year ice habitat and biodiversity, including many ice-dependent species who may use this region in future as refuge. It also, do that, that's better? Okay. It also includes many fjord systems that are heavily influenced by the ice export and freshwater export from the Central Arctic Ocean and the Ellesmere Island ice shelves. Our DFO objective is to collect baseline data to help with the characterization of this ecosystem to support future conservation strategies. And today I would like to compare two ice-covered fjord systems that we recently visited and that are located on the opposite sides of Ellesmere Island. The Archer Fjord Lady Franklin Bay system, which I now will just call Archer Fjord, was visited in September 2023 during the onset of the sea ice formation, but we also encountered a lot of exported sea ice at the entrance of the fjord. The Nansen Sound Greely Fjord system, which I now just call Nansen Sound, was visited in September 2025, so very recently, also during the freeze-up period. And there, during our 2-week stay, we watched the ice grow very rapidly around us. And those pictures, they are just, I find it stunning. So both surveys were part of a larger expedition, or two larger expeditions on board the Canadian Coast Guard ship Amundsen, and represented the first comprehensive oceanographic studies of these areas. We tried to capture a snapshot of the entire ecosystem, including sea ice, the pelagic, the benthos, the sediments. But today in this short talk, I will only focus on the sea ice conditions, the water mass characteristics, and patterns in primary producers. As I mentioned before, I didn't actually mention it, but there is barely any data available for both systems. The bathymetry is largely unknown. While we were there, we discovered sills and shallow areas, but sound is deeper and shaped like a deep trough. Arctic water exits Archer Fjord through Narrow Strait and Nansen Sound through Eureka Sound, that's highlighted with the black arrows. Both systems receive a large amount of fresh water from atmospheric deposits, sea ice that locally melts there, and the Arctic Ocean. Archer Fjord also receives fresh water from Lake Hazen through Ruggles River, and Nansen Sound receives large amount of fresh water from the Ellesmere ice caps and 3 marine terminating glaciers. The ice cover in Archer Fjord is generally dominated by multi-year ice, which briefly retreats in summer. The ice cover in Nansen Sound is dominated by first-year ice, which almost fully melts in summer. And in Nansen Sound, there's also an ice plug forming at the entrance, with influences the sea ice export into the area. To learn a bit more about the sea ice conditions in Tuvayutuk, we further investigated historic trends in an ongoing, under review publication. And for that, we used publicly available weekly ice shots from the Canadian Ice Service. And before I go into detail, I would like to highlight that Nansen Sound is 9 times larger than Archer Fjord, which explains some of the more drastic shifts in the dominant ice type we observed. So those plots show the average extent of first-year ice in purple and multi-year ice in orange. And in Archer Fjord, multi-year ice wasn't always present in winter. So these plots, they also show the maximum ice extent concentration of the different ice types in April between 1999 and 2020. So here you can see multi-year ice is not always present in winter, but our analysis didn't show any significant trends. In Nansen Sound, we did see a significant increase in first-year ice by 30% and a significant decrease in multi-year ice by 59% in April. And in 2020s, and a couple years after, multi-year ice was virtually absent from the system. The next plots show maximum extent of open water between July and September for the same time period. In both systems, we saw significant trends in the positive trends in the maximum extent of open water and the length of the open water period. And these plots also show that both systems become more often either nearly or fully ice-free, which highlights a shift towards a fully seasonal ice cover in both systems. With the CTD data we collected, we were also able to characterize the main water masses. And in Archer Fjord, we saw a fresh polar mixed surface layer near the freezing point. Pacific water was also present above 150 meters, and below we detected Atlantic waters. In Nansen Sound, we saw the same water masses at the surface near the freezing point, but overall, the surface layer of the first 50 meters was much fresher. We also observed a temperature maximum at 10 meters in the smaller fjords, which may be a remnant of summer solar heating that's now kept by the fresher and colder surface layer in winter. And a recent study by Dimitrenko et al. Also showed that there is an increased presence of Pacific water in the intermediate layer by comparing our recent CDD measurements with data from the 70s. Because I am interested in Arctic primary production, I also want to give you a glimpse into the phytoplankton biomass that we measured. And overall, we saw very low integrated biomass measurements that are comparable to measurements in the Central Arctic Ocean. In Archer Fjord, we observed a nitrate-limited fall bloom, with chlorophyll A concentration being highest near or at the surface. In Nansen Sound, we only saw shallow subsurface chlorophyll maximum with an overall much lower chlorophyll A concentrations. Only in the small fjords and in Eureka Sound, we saw similar chlorophyll A concentration than we did in Archer Fjord. Here, nutrients in the surface layer were depleted to much deeper depth, to 30 and 40 meters, which might point towards a prior high consumption. And this conclusion of a possible post-bloom stage in Nansen Sound matches our observation with a much higher dinoflagellate contribution to the overall phytoplankton community in Nansen Sound. But in both systems, biomass was dominated by diatoms. So to kind of conclude with what we saw, that environmental drivers seems to differ that control primary production. In both systems, we saw a strong density-driven stratification with limited mixing. But in Archer Fjord, freshwater gets flushed out much faster by narrow strait, which leads to more variable nutrient concentrations. In Nansen Sound, it acts more as a closed system that accumulates freshwater over the melt period, and this leads to an enhanced stratification, which may limit the nutrient replenishment of the surface layer year-round. Based on our observations on a fall bloom, Archer Fjord shows the potential of 2 annual blooms in the short but expanding open water period. And we also saw from our sediment analysis that the organic matter accumulates in the benthic over long term, which acts as an additional food source for the benthos. But interestingly, we saw overall low biomass in pelagic and benthic communities, which doesn't really help us explain the known hotspot for marine mammals in Archer Fjord. So we see a lot of walrus, narwhal, and seals there. And this productive paradoxum, to steal Max's words, needs to still be solved. In Nansen Sound, we saw overall biomass, low biomass of the entire food web. But we did see local spikes in chlorophyll due to local upwelling events. So our next steps will be to include data from satellite observations and moorings that we deploy to get a better idea about the seasonal dynamics. And with that information, we can hopefully better understand the presence and absence of fish and marine mammal species, but also it will help us to further characterize key ecosystem risks. If you want to know more about our observations, some of it has already been published and lots more is to come over the next years. And with that, thank you.
question? If there are any.
Yeah, thank you for the really nice talk. So you said the open water period is expanding. So what is actually the duration of the ice-free period, and like what's the difference between the spring and the fall bloom? You said there is sometimes two.
Yeah, so in Archer Fjord, the open water period ranges about 30 days. In Nansen Sound, it's a bit longer. And for Archer Fjord, the bloom characteristics, we don't have any observations of a summer or a spring bloom because it's really hard to get there. And September is kind of the only time where Coast Guard is willing to try to go there. So we see that the biomass is very low still, signal in the surface. So we still need to figure out if there is a spring bloom, how big it is, can it support this food web? It's a huge question we try to solve.
Could that be the spring bloom, the one you see in September, essentially? Since
Personal opinion, I don't think so because there were 30 days beforehand where things could have happened. Okay, thanks.
Thank you very much. Our next speaker is Elena Eriksson. She's based at the Institute of Marine Research here in Norway, but also affiliated with the Polar Branch of the Russian Federal Research Institute of Fisheries and Oceanography, or PNRO. Her talk is about the living Barents Sea adapting to climate change.
I am not. I am actually from Institute of Marine Research.
Okay.
Yes. Today I would like to present a paper that was published last year, and the study was done by former and current members of ICES Group IBAR, Integrated Assessment for the Barents Sea. Motivation for this study was the Barents Sea has experienced warming, and especially a strong increase in temperature. I come very close now. In temperature occurring between 1980 and 2016. And therefore we will study how living Barents Sea responded to this strong increase in temperature. We investigated abundance and distribution of living marine ecosystem components and how they respond to climatic variation. And whatever possible response was great in the area with strongest oceanographic changes, dependent upon the species trophic growth and causing ability of the system to recover when the influence of external forces ceases. We have used data from the comprehensive Barents Sea ecosystem survey in August-September in the period from 2005 to 2022. The living Barents Sea represented by more than 200 species that we combined into 19 biotic groups and 6 assemblages. We calculated 299 time series that help us to assess the response of the ecosystem components to the climate change. We also want to see a spatial variation, and we divided Barents Sea into the polygons. You see these polygons, and the main currents, warm Atlantic water entering to the Barents Sea from southeast, and Arctic cold water entering from the north. And of course, they influence the thermal condition in the Barents Sea, in the different areas. Temperature constraint cluster analysis of the temperature above 100 meters shows that warming was not continuous over the study period. Yeah, but it could be divided in 3 periods with different thermal conditions across the Barents Sea. The period from 2005 2005 to 2022 can be divided in three. With bars, you can see annual anomaly for entire Barents Sea, while with lines for each polygons. And the first period from 2005 to 2011, it was cooler than the whole study period, followed by a very warm period from 2012 to 2016, and finally a cooler period again from 2017. If we look at the spatial distribution of the average temperature, with colors you can see anomaly, with numbers it's average temperature for the polygons. And during the peak warming period, whole Barents Sea become warmer. And it's the most significant warming occurred in the northwestern and southeastern polygons. Here is a temperature from 10 to 100 meters, and there's a bottom temperature. It's similar patterns we see on the bottom. During the cooling period, almost all polygons become cooler, but North Svalbard and Pechora, they become even warmer than during the peak warming period. It means that the warming continues in these two polygons. When we look at how Barents Sea biotic responded to climate change, we see that the response was different from different ecosystem components. For example, mesoplankton. It's declined, but it was not directly from warming, but it most likely was due to increased predation from a large stock of capelin and increased abundance of herring. Opposite, jellyfish, they expanded the spatial distribution and increased in the biomass. This indicated favorable condition in the entire Barents Sea. Demersal fishes that were dominated by cod, both juveniles and adults, they decreased in abundance following the cod stock decrease. Small Arctic species in the northern Barents Sea returned during the cooling period, likely driven by ice retreat and reduction of predation pressure from declining cod stocks. Snow crab spread over the larger area and increased in the biomass. When we look at how biota in the different area responded to the climatic changes, we see that the Svalbard North, near the Svalbard, a large increase in biomass of almost all components, from macroplankton, juvenile fish, demersal commercial fishes, large demersal fishes, while in the south, where is it? While in the southeastern part, the already low biomasses of zooplankton and fish in the region has become even lower, while jellyfish and snow crab have increased significantly in abundance. And here we see that climate change, very strange. Climate change led to ecosystem change in the southeastern Barents Sea. Take-home message. The ongoing warming in the Barents Sea is not linear, but rather occurs in different phases and varies spatially. The strongest biotic response occurred in the areas with strongest oceanographic changes. And climate change led to ecosystem change in the southeastern Barents Sea, resulting in dominance of jellyfish and crab, and a reduction of fish and plankton. That will have, of course, consequences for the rest of the ecosystem. And I will end that such changes are challenging to access and predict without a comprehensive ecosystem monitoring. Thank you.
Thank you, Elena. We'll have time for one quick question.
Everything clear.
That's okay. Good.
Yes, thank you. Next speaker is Edda Johansen from Institute of Marine Research. She will present poor recruitment of Northeast Arctic cod, evaluation of potential causes.
Okay. Good morning. You already said the name of the talk, so you saved me some minutes there. So as you all know, this is cod, and it's one on shelf species in both sides of the Atlantic, it's a demersal fish. And it has a very long history of exploitation, so it's a very important commercial species. These two graphs shows the landings of cod in the northwest and northeast Atlantic from 1950 to 2023. And you can see how in the West Atlantic the catches have plummeted due to stock collapse, and there are no commercial fishery there now. In the northeast Atlantic, we are doing a bit better. So in blue, you can see the northeast Arctic cod, which is dominating. In red is the Icelandic cod, and in green, all other stocks that are found in the eastern Atlantic, and some of them have been reduced. So currently, the northeast Arctic cod, or historically also, at least back to the 50s, it's the largest stock in the world, and it lives here, a bit north of us in the Barents Sea, and it spawns south of us in the Lofoten area. That's the main spawning area. So given its importance, it's very important that, of course, we manage it sustainable, and then we have to understand the dynamics. This plot shows the stock dynamics, the development of the stock. So it's the biomass of the spawners in blue. And unfortunately, the stock is in decline. And the spawning stock biomass is now reaching a critical level, the B-lim, that's a purple line. So that means it's reaching the level where the recruitment is severely impaired. And more specifically, the recruitment success, that is the number of recruits divided by the spawning stock biomass, or the number of recruits produced per biomass of spawners, has dropped abruptly after 2006 and remained low thereafter. This is the average level. There are some variation, but the level has dropped. And together with the fishing pressure that has been relatively high, at least in some years, this has led to the stock decline. This shows the life cycle of the cod. It's a very thoroughly studied stock. So all these things that are written on this pie chart are factors that impact recruitment of cod. And we have made a review paper that is in review. And we asked if all these studies that's been done could inform us on the reason for the poor recruitment success that we have observed. So cod spawns in these purple areas on this map along the coast of northern Norway, whereas the main spawning area is around Lofoten. And they spawn over an extended period, but the peak spawning is in early April. And then, depending on the temperature, they will hatch. The egg will hatch after 2 weeks. And then they will live pelagically for half a year about. So the eggs and larvae are transported into the Barents Sea by the Atlantic current, shown as red in this map. And in September, half a year after they are been spawned, they found over wide areas in the Barents Sea as pelagic juveniles. That's what's shown in pink on this graph. And then during the first fall, they will settle to the bottom and start their demersal lifestyle. And they recruit into fishery when they're 3 years of age. So on this map you show in green, that's the distribution of the same year class. This is the 2008 year class as 2-year-olds. So they changed their distribution quite a bit from their pelagic zero groups till they are 3-year-olds. So we have an abundance index from September when they are a zero group distributed in the Barents Sea. And from this, if you use that as an index of recruitment success, there are no really abrupt change. So it's a lot of variation, but the number of zero group produced is quite proportional to the spawning stock biomass. Whereas when you use the H3 as a recruitment index, you can see this changing level. So we think that the recruitment failing happens between when they are half a year and are pelagic and until they are recruited into the fishery at age 3. So there seems to be reduced survival from their pelagic juveniles when they are half a year until they are 3 years of age. The question is why? And spoiler alert, we don't really know why. But we have some clues. So we have observed reduced inflow from the Atlantic water into the Norwegian Sea, or that was not what, from the Norwegian Sea into the Barents Sea. And that brings copepods into the Barents Sea, and this is important food for juvenile cod, and also for cod's main prey when it is a bit bigger, capelin. So despite reduced survival. And then also what has been observed is a northward shift in spawning grounds. So Lofoten area is a traditional fishery area for centuries. And we have a survey there at the IMR Institute of Marine Research. And last year there were practically no fish in the Lofoten area caught on this survey. Of course, the stock size was low, so that's partly the reason why, but they caught spawners further north, so they seem to have shifted their distribution further north. So why is this important? Modeling studies have shown that where they are transported as larvae and eggs into the Barents Sea depend on where they are spawned due to how the currents go. So one possibility is that they settle in unfavorable areas. And Elena has shown that, for instance, the Eastern Barents Sea has changed quite a bit. So if you see on this map, you can see a lot of fish in the southeastern Barents Sea as 3-year-olds, and maybe this is an area where the survival has been reduced because of changes. So this is something we will look more into. So to conclude, it seems to be poor survival from zero group to age 3. And I haven't mentioned that in particular, but the first winter seems to be a critical period. That's they have to have enough resources to survive this winter when it's dark and not much food around. There have also been changes in Atlantic inflow with less zooplankton. And they seem to be spawning further north and east, which might lead to juveniles ending up in what I call the wrong areas. High temperature, I haven't really mentioned climate change. High temperature seems not to be a problem in itself, as of yet. No boiled eggs. But of course, there are indirect effects. But anyhow, when we have this situation, this poor recruitment, it's very important to reduce the fishing pressure accordingly, so we don't end up in this situation, which is something we don't want, of course. Thank you. And I want to thank also Husebø, who made these nice graphics. Yeah.
Yes.
Thank you for a very nice presentation. You mentioned explicitly that the larvae seem to be maybe very vulnerable. My question is, might there be some mismatch with the potential slightly earlier hatching or spawning with the accessible food? And even, I think in lab at least, we've seen that the cod might have a green gut for a short while, so they may need that primary production that might come later with the light.
Yeah, the thing is that they seem to be doing quite well up until the zero group stage. Of course, it could impact their condition that they're in when they enter their first winter, but we don't really see the effect in a zero group. There's no reduction in a zero group, so that's our main conclusion.
One last question.
Thanks a lot, Edda. Really interesting. I'd like to follow up on the question regarding body condition. So mortality likely in that juvenile phase until age 3. Do you also see a strong decline in body condition of those juveniles, suggesting that there might be food limitation playing out?
I looked at Elena and she said no.
No. Okay.
Yeah.
Interesting.
Yeah. But there is a project ongoing to look at more like the fat composition of zero group. It's been explored. Yeah.
So starvation might not be the most likely. What about parasites?
Really no.
Well, continue to puzzle us.
Yeah.
Critters.
Next speaker, Melissa Ciretti from Institute of Marine Research, and she will present Arctic Ocean acidification trends and effects of progressing climate change. Please.
Thank you very much, and I also added that I also want to show some of the recent studies on some of the biological impacts. And I also want to thank Agneta Fransen and Elizabeth Jones, which also contributed to this work. And you know, as we have seen previously, that the physical state of the ocean is changing with warmer temperatures as well as sea ice loss in the Arctic. But we also know now that the chemistry of the ocean is changing. And the increased CO2 in the surface ocean has led to a less basic state in the carbonate chemistry, meaning that we have decreased pH, but also decrease the carbonate ion concentrations. I'm just trying to find the laser there. So this iron here is controls the solubility and saturation of minerals, calcium carbonate minerals such as aragonite and calcite. So the lower aragonite saturation, which is depicted here as omega aragonite, means that the organisms needs more energy to form shells and skeleton. At the aragonite saturation below 1, there is a risk of dissolution. It's a chemical limitation, but usually the organisms needs higher omega saturation than this. On this map here shows the saturation change from the pre-industrial values, 1750, to the current state. And as you can see in the Arctic, we have the deeper red and brownish areas shows that we have had a saturation decreasing by 30%. And we have looked to different studies, and I simplified some of the studies because it's horrible to say a change in pH. So I summarize it easily like this, but we know that acidification is occurring between 2 to 5 times faster in the Arctic. And it's on the Pacific side, it's about 5 times as fast rate, and also in the Arctic outflow water on the Western Fram Strait, we have seen faster rates. but also considerably faster rates in the northern Barents Sea, in the Arctic domain, as well as in the Nansen Basin. And these are some of the intervals that you can see here, the change per year in some of these areas. So the reason for this fast acidification is climate change. So in the Arctic Ocean, we now see a freshening, so more river runoff, but also increased sea ice melt. And this fresh water is diluting these carbonate ions in particular, so enhancing acidification. But also, this fresh water is also changing the surface ocean towards a more soluble. So you have cold and fresher water, which has a potential to take up more carbon dioxide from the atmosphere, in particular now as we see more open areas in the Arctic, and we get enhanced CO2 uptake here. So there are also some recent studies that are really trying more and more to connect the chemical change to biological impact. So by using experiments, but also values from biological assessments on different marine organisms and groups, together with the changes in the carbonate system, trends and ranges of pH, for example, and aragonite saturation, together with models, one has succeeded to at least suggest some of the thresholds and tipping points based on a collective combined information from these studies. And particularly vulnerable species are the polar pteropods, the Limacina helicina, larval stages of bivalves, gastropods, decapods, and as well as corals. And some of the tipping points that has been suggested is that for the pteropods, you can see shell dissolution already occurring at aragonite saturation below 1.5 and severe dissolution at 1.2. And bivalves is less than 1.9. So if we go to the Arctic, this study also combined models and observation, and this shows the aragonite saturation in the upper 200 meters, and the more red numbers you see, that we also the lower aragonite saturation, and in the Eurasian shelves as well as in the Beaufort-Gyre and Canada basin, you have very, very low aragonite saturation levels. They also then looked into the percentage of dissolution of pteropods in this combined model and observations, and they could see that in these areas it was more than 80% severe dissolution of the pteropods. But this also means that when you lower the aragonite saturation state, you also increase the area where you have aragonite saturation lower than 1. So it's been, since the pre-industrial times, it's been a 21% increase in the area where you have a dissolution. When we also take into account the thresholds, we see that the suitable habitat for pteropods has decreased by 61% and for bivalves by 13% compared to the pre-industrial values. Another study was also showing the carbon dioxide level and the tipping point, ecological tipping points caused by high CO2 levels. And they also looked at in particular, physiological changes. And in this table, they summarized for the impact from different organisms. And in the red areas is a negative impact, and the values in the squares shows at what PCO2 level this impact occurred. And for example, for calcifying phytoplankton and coralline algae, the tipping point was starting at 538 ppm. And they also see that the acidification will, in near future, impact the most benthic marine ecosystems, and also at tipping points as low as PCO2 levels at 500 microatmosphere. So in a way to trying to put these thresholds, tipping points, and these values into context, we can take a look at the bottom water values in the Barents Sea. And as you've seen from the other, we have this North Atlantic warmer influenced area in the south, and the Arctic domain in the northern part. And here is the aragonite saturation in the bottom water, and the more purple than blue, the lower aragonite saturation. So we have already values that are lower than 1.2 in the bottom water of the Barents Sea. So also areas where we have the lowest pH, but also the highest PCO2 levels, and they are more than 450 ppm. So it seems like we might already have reached some of these tipping points that they suggest from these studies in the Arctic domain of the Barents Sea. We should also remember that with thinner and lighter shells, we'll decrease the carbon export to the deeper layers. So it means that the food source to the benthic communities will decrease, but also that the carbon sequestration will also decrease. So the ocean will not be able to pump down as much carbon dioxide or carbon to the deeper layers to bury it. To summarize, so the Arctic Ocean acidifies up to 5 times faster due to climate change. Shell dissolution already impacted pteropods bivalves. Safe space for Limacina helicina has decreased by up to 61%. And we should also remember that acidification is a compound effect of many different stressors and pressures, such as increased temperature as well, really enhance acidification effects. And we hope also to close some of the large knowledge gaps in the Central Arctic in this new project, Arctic Ocean 2050. And I want to say thank you, and this was supported mainly from the project Sudarko and Clean within the Front Center project. I did it.
Any questions?
I'm Misha from IMEDEA, Spain. Thank you very much for a super nice, inspiring talk. As I saw from one of the first slides, there is a positive effect on calcification for fish and negative for bivalves. Yeah, so there's
Yes.
It's directed differently. So how could it potentially impact the consumer of this prey, for instance, walruses? Would it make bivalves more profitable and fish less profitable for them? Would they change their diet? So
yeah,
if you
no,
answer.
we have an example from the Pacific Arctic in the Chukchi Sea area where they actually seen that bivalves at the bottom have had shell dissolution, so impacting the I think the diving the aders, but also the walruses because they mainly feed on the bivalves. So it might have large implications for sea mammals.
Thank you.
One last question.
Thank you. You mentioned that Limacina helicina will be heavily impacted, but what about retroversa, which can be transported to the Arctic Ocean?
I think there have been less studies on the retroversa, but they say polar pteropods. So I think we have less information on retroversa, but I would assume that it would also be impacted because retroversa is residing more in the Atlantic water, or we find it in the Norwegian Sea, where in a way you have more, the carbonate chemistry is more favorable than on the Pacific side. So if the retroversa would enter into the Arctic area where you have higher dissolution potential, then I think also retroversa would also be impacted negatively. But I haven't seen any studies. Yeah, that's the thing with the different feedbacks. As you say, it's both these green areas and the red areas, but some studies also say that there will be more jellyfish with acidification because they don't have this calcium carbonate shell, some of them at least.
Thank you.
Thank you.
Next speaker, Pedro Duarte from Norwegian Polar Institute, and he will present Atlantification and contrasting fjords environments. Insight from Konsfjord, Ripfjord, and Svalbard.
Okay, so I will say a few words about based on the contrast between two fjords. And as most of you know, fjords that are located along the West Spitsbergen, they are pretty much under the influence of warm Atlantic water, and this effect, this warming effect, due to these exchanges, adds to the increases in air temperature. So it's a kind of cumulative effect. So we compared Kongsfjorden, which is a relatively warm fjord and very well studied, with Ripfjorden, which to some extent is still a cold Arctic fjord. And this figure shows you the pathways of Atlantic water in red and the pathways of Arctic water in blue. We hypothesized that differences in fjords, such as these two, in their water masses, prokaryotic communities, they may provide some insight about the effects of Atlantification as it expands north and eastwards, because there is evidence that Atlantification is getting stronger and stronger, and that in the future, probably, Ripfjorden will become more and more affected by it as well. So the methods, basically, this study was based on transects done in Kongsfjorden. This is just a detail of the stations inside the fjord, and also in Ripfjorden, along these stations, so from the inner part of the fjords until the deep ocean. And these transects, they were done in the summers of 2012, 13, 14, 16, and 17. We collected physical and biogeochemical data. And in these two last cruises, we also collected samples for genomics and metagenomics. I'm going to show you only a very small subsample of the results, and then, depending on the questions, I may tell you more about the others. So here, I'm showing you two columns, one for Kongsfjorden, another for Ripfjorden. This axis shows the distance from the inner fjord stations, and this is the depth. And here I'm showing you only a part of the transect for visualization. The color scheme represents different water masses, and they are identified, I mean, the color with water mass at these four bottom panels. So we have Arctic water, Atlantic water, intermediate water, local water, surface water, and so forth. And the isolines, they represent nitrite plus nitrate concentrations. And the main findings from these results is that the fjords are dominated by different water masses. This is no big surprise. They actually show some similarities in 2016. As you can see, for most of the time, we find cold water masses inside Kongsfjorden, such as Arctic water, local water, and winter cold water, whereas in Kongsfjorden, we find transformed Atlantic water and Atlantic water. But in the 2016, there is quite a lot of similarities between these two fjords. We also see that the nitrate isolines, they tend to be tilted as one move from the shelf to the fjords, and this suggests that the fjords are removing nutrients from the water. We proceeded further to study better what is happening with nutrients by grouping the fjord stations separately from the shelf stations, and we did this for Ripfjorden, for Kongsfjorden as well. You can see here the two groups. And basically what we did next was to apply mixing diagrams to try to separate, the effects of dilution as the waters enter into the fjord and mix a bit with fresh water coming from the glacial drainage, from the effects of biogeochemical sources and sinks. And we came up with these results that I show you for each of the years when we did the sampling. So here you have the amount of nitrite plus nitrite that was removed or added to the water by biogeochemical processes. Negative values means sinks. So basically what I'm showing you here are medians and ranges for the different years for the different fjords. Kongsfjorden with warm colors, Ripfjorden with blue colors. And as you can see, the results between the two fjords are quite different. And the main take-home messages are that Kongsfjorden is consistently a nitrogen sink. Ripfjorden surpasses Kongsfjorden as a sink only in the warm 2016. Other than that, you have here points that are generally lower than those for Kongsfjorden or close to zero or even positive. Now, moving now to the genomics, here we are contrasting Ripfjorden with Kongsfjorden and the 2 years. We have, as I said before, regarding genomics and metagenomics, and I will not talk about metagenomics today, just now. We have data only for these 2 years. And then you can see here we have the depth in the log scale and the number of amplicon sequence variants in this axis, showing basically the alpha diversity for each year, each fjord. And the color scheme represents the different water masses. And the main take-home messages are that alpha prokaryotic diversity increases with depth generally in both fjords. The highest values typically are found in the deep winter-cooled water. And we have higher prokaryotic diversity in Kongsfjorden. So the warm fjord has systematically, even for comparable water masses, higher alpha diversity. Now, focusing on the genera associated with the nitrogen cycle, here we represent in the red and orange shades ammonia-oxidizing archaea, dark gray for ammonia-oxidizing bacteria, and green for nitrate-oxidizing bacteria. And the light gray shades were used for the proportion of other genera that are not related with the nitrogen cycle. And you can see clear differences between fjords and also between years. There is quite some internal variability. But the main take-home messages are that N-cycle-related prokaryotes were more abundant at near bottom depths, both in transformed Atlantic water, the deep layers of transformed Atlantic water, and especially winter-cooled water. And we also see that archaea were systematically more abundant than bacteria and nitrate-oxidizing bacteria as well. So as final remarks, Kongsfjorden and Ripfjorden showed contrasted hydrography with stronger Atlantification in the former. Warmer Atlantic water influenced waters were linked to enhanced nutrient uptake and sink potential of these fjords that we confirmed for other nutrients and for dissolved inorganic carbon in the case of Kongsfjorden as well, with data from Melissa and Agneta. But this was already published. Distinct water masses supported distinct microbial communities with higher diversity in the warmer fjord. These results reveal system-wide impacts of Atlantification. I mean, they allow us to start to foresee a bit more how Atlantification will impact fjord biogeochemistry and microbial communities that is intimately associated with all the ecosystem functionings in terms of sink source for nutrients and carbon. We argue that it's very important to keep monitoring and that these monitorings should include the marine end members of the fjords, not being focused only inside the fjords, but also on the shelves, so that we could contrast local with regional process. Thank you.
We have time for questions. I have one. So the Kongsfjorden and Ripfjorden are common fjords to be contrasted. Is there a plan to maybe compare that to other regions of the Arctic also? How do you think that translates to other regions of the Arctic that have similar identification versus more typical Arctic conditions? Yeah. So my question is, how representative do you think these changes, contrasting between Kongsfjorden and Ripfjorden, are compared to other regions of the Arctic?
You guys are not deaf, so you don't need this. But I mean, we actually took part in a project, in the European project, Face It, that ended last year. And we were supposed to extend these comparisons to many fjords, but the people that were in charge of doing so, when they started to assemble data, they came up with the conclusion that it was almost impossible to find a common and representative data set that we could use to cross many different fjords, at least in the European Arctic. This was quite frustrating, actually.
That's why you need to compare also in Canada. Thank you. Good, thank you.
The next speaker, John from Alfred Wegener Institute, presenting a risk and challenges posed by the increased potential for harmful algal blooms in the Arctic. Please.
Thank you. Yeah, hello everybody. It's nice that I have the chance to present our data here for my two biggest projects. It's Fjord Change, or Polfjord, and Greenhab. And you see this with many collaborators. At the end, I have a picture of many, but it's never complete. So, well, so first of all, I think this I can skip. We all agree we are in the Anthropocene and things are changing. And this has, of course, a huge impact. And in my studies and these projects I'm presenting here, it's about the impact of climate change, so ecosystem structure and functions. Now we look into the recent part and in the past of plankton communities. So in the past, we go for sediment cores and look into the diversity. In here, and this is also what I present, we have an emphasis on HEP species, so harmful algae bloom species that can affect the ecosystem. And we try to understand the risk for the Arctic. Okay, harmful algae blooms, as I already, the names or harmful algae, they affect the ecosystems. And this can be very dramatic, but they also can affect the human health by, for example, consuming contaminated seafood. And I give you here 3 examples, which are quite prominent. This is one study. There are many of this group from Don Anderson and colleagues. This is Alexandrium catenella. It produced saxitoxin, a paralytic shellfish poisoning, which vectorized through the food web and can contaminate shellfish. If this is consumed, you can become seriously sick. And they found in the Alaskan coast here a huge cyst bed of this resting stage over the winter time. And this has, it's still too cold to hatch, but they're fertile. So if the groundwater would become warmer and warmer, there would be a serious risk for this area. Then there's a study again in this similar area where they have from Pseudonychia, a penadiatum, a toxin called domic acid, is a neurotoxin. And it's also a movie star, if you like. This was also the vision for the film from Alfred Hitchcock, The Birds. So it makes, it vectorizes over small crab and fish, and quite a bit of mammals, but also birds when they get then crazy and affect people. The other story I want to introduce is what was here in this area, so that's why I choose it. This was a bloom 2019 of Chromichromelina ledbertii, where you had a huge loss in the salmon farming industry here. I just learned this was about 2% of the annual money they make, so it's a huge impact. And yeah, so we and others studied this a lot. And so if you want to have more results about this, we can talk later. So what we do is, if you look in the ecosystem, we think ecosystems are very complex, especially plankton dynamics and species interactions. So what we use is, we look at the dramatic chaotic soup. We use now molecular approaches to reduce the complexity into ACTG, there's only 4 letters, or we use parameterization of our metadata to numbers, and then this becomes a bit more organized, and simple minds may understand this. So when I show you, now I show you a few examples of species that may have moved to the north, which are relevant in this case, right? You see the fish-killing ones, like I show you an example of Pseudochatonella. You find them here in this area, Barents Sea, but also around Svalbard. We found them also in Greenland. Chrysochromelina, I talked around already about it, but also Promnesium polylepis, you may know from the 90s, or Promnesium parvum, they found here in the Arctic already. Another example is Pseudonychia, our film star. So you see it's also in this area. We have it around Svalbard, and it's also very prominent in other regions in the Arctic. And here, due to new times, we understand the biosynthetic pathways. For some algae we do, so we know how they are produced, and then we can also target genes. And you see here an example where we found active expressed gene signatures in Greenland, Svalbard, Iceland, and Norway. And that helps us a lot. So not only detecting the species or the toxins, but when we see the activity of the genome producing the substance of interest, that explain a bit better the risk and the dynamic of the bloom. The same we can do for Alexandrium, as I showed you before. So the question we have, we see that they are there. The question is now, are they coming only by currents in spring and summer, or are they coming to stay? So do they establish, or have there been already established population or within the area? And then understanding the impact on the food web, human health impact, and socioeconomic consequences, which is of course very dramatic in areas like Greenland, where the economy is nearly 100% dependent on the sea and fishery. And therefore we have the project GreenHab. And in this GreenHab, this is a German government-funded project where we have a complex interaction of many work packages, but in principle it goes around the distribution of harmful algae, the evolution of the species, how good they can adapt, then the competitive ability, how they can do this. We look at evolutionary traits to see look for local adaptation, and we do some modeling to see how things are changed, because we need to understand indicators we can work with and tipping points to see when things are changing. And finally, we put it at the end, but it should be at the start. We have a dialogue to the Greenlandic communities. And this is also how it started, actually. So I give you a short introduction to the different work packages. We have results, but there's too many, and I hope to catch you up, or you catch me during the coffee break, and then we can discuss it a bit more in detail. So in work package 1, it's about the assessment and evaluation of the connectivity, so who's there and how they are connected. Therefore, we use a classical barcoding approach, take the community, sequence them, we get barcodes, and can assign them to species, and we can show you that we find all over the year toxic algae around Greenland, so they are already there, right? And with this work package, we use a metatranscriptome, so that means the active genome of the community, not the DNA. We take the RNA, and this is a bit like the discussion of the genome of the system. And we filter out the information we get from the toxic species and look for certain functions and processes involved in local adaptation. How much species are adapted to the area, or what makes the local community more resilient to the area, or what makes the local community more resilient. In this work packages, it goes about the adaptive evolution. So what we try to understand is what are the mechanisms of the local adaptation. So what makes the local community resilient, and how toxic temperate species, different environmental drivers to become more competitive against the local communities. So it's a long-term evolutionary experiments, and then we put this in the next work package, non-HAB species and community and single ones with the HAB species together. And in the first place, the non-HAB species win in the given condition. When we change the conditions with the different drivers, we see that the outcome change. In some cases, the local community is still resilient, but in other cases, we get coexistence. But we have also some. And this is tricky, and we need to understand, because that could explain tipping points when this species will be established and dominate the new communities. Yeah, and in work package 6, we do a HAB modeling for the Arctic. It started with a food web model where we add harmful algae and non-harmful algae. What is very important, we need to add competition, grazing, copypod grazing seems to be very important, and mixotrophy. Bringing this together seems to drive the model in the way that we can look for some conditions where harmful algae may become more prominent. Yeah, and as I told you, we started with work package 7. So before the project started, in the first moment, we hired Maurits. We went in the first 6 months several times to Greenland to get into contact with the communities, local authorities, to really understand what kind of question and results they want us to produce with them together, that it's meaningful for the Greenlandic society. And finally, we did quite a bit interaction with gymnasiums, like open ship days when we had a cruise where we invited the people I got it, and we worked with a university and produced in Danish and Greenlandic and English some information sheets they can use for education. And I'm running over time. If you want some more information, you have here the webpage, and I'm hanging here around for answering some questions. Thank you very much.
Thank you. We'll take one quick question, if anybody has one. Thank you very much.
All crystal clear.
Yes, coffee break. So thank you very much for joining this first part of the session on climate change and cumulative impacts on ecosystem. I made a mistake at the beginning. It's actually 4 parts. So this afternoon we have 1 part on cumulative impacts. Then we have central. There's the co-creation of knowledge. 5 parts with the panel afterward. Yeah. Yeah. So there's more than, and we invite you to join. So we'll see you there. Thank you.